The overall goal of this procedure is to disrupt the blood-brain barrier. This is accomplished by first placing the animal on IV anesthesia with an endotracheal airway. Next, the bifurcation of the common carotid artery is exposed. Then a catheter is inserted into the external carotid artery. Finally, Mannitol is administered. Ultimately, results can be obtained that show blood-brain barrier disruption through extravasation of Evans blue dye.
The main advantage of this technique over existing methods of intra-arterial therapy is it allows drugs to enter the brain which do not ordinarily cross the blood-brain barrier under physiological conditions. This has implications in the field of neuroscience because it allows us to test new therapies which have poor ability to cross the blood-brain barrier. Implications of this technique extend to neuro-oncology because many chemotherapeutic agents do not cross the blood-brain barrier well.
Though this method can provide us insight over neuro-oncology, it can also provide us information on other neurological disorders treated by drugs with poor blood-brain barrier permeability. We hope that the visual demonstration of this method will allow researchers unfamiliar with animal surgery to use this technique in their labs.
To begin, prepare a solution of 25% mannitol and verify that there are no crystals. If crystals are present, dissolve them by placing the vial in an 80°C water bath and agitate periodically. Record the mass of the animal and prepare two 12 cc syringes of propofol